Insect pests cause serious damage to important cultivated crops. In the last few decades, chemical control has been widely adopted for the management of these insect pests. However, dependence on pesticide is not sustainable because of associated environmental problems, development of resistance to insects, insecticides, and an outbreak of secondary pests. Hence, there is a need to identify suitable alternative methods for the management of insect pests. What is white grub and why is it dangerous? Grubs are among the most destructive and troublesome of soil insects, threatening the entire crop production, nullifying the gains of high-yielding variety programs in the country. White grubs are a pest of commercial crops. The white grubs are also called root grubs. White grubs are among the most destructive and troublesome insect pests all over the world. White grubs are also called national pests. Their infestation has been reported throughout the country and the magnitude of the problem has been widespread over the past years. In the majority of the farming situation, control of these pests is important because of the lack of control over their damages. White grubs feed underground on the roots of host plants, while the adult beetles are observed feeding on the foliage of certain other choice plants in the vicinity during the night. The damage done by grubs to agricultural crops is sometimes more than imagined. The losses inflicted to the various crops by this pest range between 40 and 80%. White grubs, host plants. The major host plants of white grubs are sugarcane, groundnut, and vegetables. The other plants like banana, citrus, pomegranate, maize, turmeric, arecanut, cardamom, potato, turf grass, and ornamental crops are also prone to white grub attack. Current scenario of white grub control. Currently, 80% of white grubs are being controlled by toxic and hazardous chemicals like chlorpyrifos, ferrate, carbofuran, and quinalphos. What are entomopathogenic nematodes (EPN)? EPNs are a group of nematodes, threads or insects which are soil-dwelling, deadly insect parasites belonging to the phylum species nematode of the Steinernma and Heterorhabditis. Entomopathogenic nematodes are families. Soft-bodied, non-segmented roundworms that are obligate or sometimes facultative parasites of insects. Endoparasitic nematodes are also called endoparasitic because they live in the parasitic form inside the infected pest, whereas plant parasitic nematodes do not kill insects but damage plants. Why is radar EPN necessary? Use of chemical pesticide in agriculture posed a concern about the hazardous effects of pesticide on the environment. There is an urgent need for methods that are eco-friendly. The development of resistance and emergence of new biotypes like EPN is another concern about using chemical pesticide, which drives us to a change in pest management methods. Entomopathogenic nematode control is an attractive alternative that is environmentally safe and feasible in crop protection. Entomopathogenic nematodes (EPN) are considered to be potential biological control agents against insect pests, and the use of EPN has now become an integral part of insect pest management. Entomopathogenic nematodes have been developed as biocontrol agents on a commercial level for the control of insect pests, especially soil-dwelling insects in an eco-friendly way. Advantages of radar EPN technology. Filled circle highly lethal to important insect pests with the least of target effects. Filled circle highly safe for humans and the environment. Filled circle no safety equipment is required, no residual effects, no groundwater contamination and are safe to pollinators. Filled circle kills insects within 24 to 48 hours. Filled circle easy to mass produce and is compatible with standard agrochemical equipment and irrigation systems for the application. Filled circle other chemicals are not required when using EPNs, they can be used with conventional application equipment. It controls white grubs, third stage juveniles of the species area infective, theory-free living, non-feeding, and possess highly sensitive chemoreceptors which can detect host cues. Venin insect has been located, Thenmatidicenta insect body cavity via natural body openings, mouth, anus, spiracles or cuticles. After entering the insect hemilymph, bacteria released produce toxins with insecticidal properties. Anti-insect jets killed within 24 to 48 hours due to septicemia, blood poisoning in the dead insect. Nematodes and bacteria feed in insect body contents and reproduce inside the cadaver. After completing several generations, the infective juvenile is released into the environment to infect other host insects and continue their life. Field evaluation of EPN against white grubs on sugarcane location percent reduction of grubs field trial 1. Ramapuram village Talavdi, Arud DT. Tamil Nadu 77.0 field trial 2. Bandahalli village Anumanthapuram Dharmpuri DT. Tamil Nadu 78.0 field trial 3. Vadakahalli village. KR Pet, Karnataka 80% field trial 4. Anikarapatti Bodanayakanur, Tamil Nadu 87.5 field trial 5. Banagahalli village Ramnagara, Karnataka 79.0.